Okay, very good. Well, I tried to talk about the work I'm doing in the U.S., and I tried to think about how I could put it in the context of South America. I've never been musseling in South America, and I don't know too much about the fauna, so I'll be interested to hear everyone's comments about how pertinent this information is to you all in your work. So I'm going to talk about the ways of monitoring mussel populations and assemblages, and also about making inferences about the health of a stream or a lake or whatever based on the performance of individuals. So I'm going to be talking about it at all three levels: assemblages, populations, and then individuals. So let's see. Okay, so why do we do mussel surveys? I suspect that in South America, there are a lot of places that maybe haven't ever been surveyed. And so, of course, that's a basic reason. In the United States and most of Canada, most of the rivers have been surveyed, and so we kind of have an idea of what species are there or have been there in the past. And so most of our mussel surveys nowadays are geared towards monitoring assemblage or population health to document declines, or hopefully document stable populations, but in many cases we're interested in documenting declines. Those surveys can help us evaluate causes of the declines and maybe recommend management actions. And if we implement some kind of management action, we would like to be able to monitor those actions and see if they're successful. So, okay, after we do a survey, how do we assess those data? Well, in the United States and in most of the world, this is kind of a qualitative assessment. We kind of if an assemblage has many species and many individuals and a lot of recruitment, and if it's increasing in size, we typically consider it's in good shape, it's healthy. And if the opposite is true for those things, we consider that population or assemblage is in poor health. And we do this kind of the same way for populations. If there's a lot of individuals, a lot of recruitment, and if it's increasing, everything's good. If it's the opposite, things are bad. But these are they're subjective assessments, and they can differ a lot between individuals, researchers, just based on their point of view and their perception. So we need to have a more objective way of interpreting mussel survey data in terms of assessing the health of an assemblage or a population. So there are a few tools available to do this in the US. There's a thing called the MCAT. It stands for, let's see, the Mussel Community Assessment Tool, and that's been published. But this was developed specifically for the Mississippi River itself, the upper Mississippi River, the main stem of it. So some of the states in the US, like Illinois, Minnesota, and Ohio, have various assessment methods or tools that they have developed for those areas. We also have some kind of indirect ways of making assessments about the health of mussel assemblages. And these are the indices of biotic integrity. It's called IBI. This is a technique. I don't know if this is used much in South America. It's used somewhat in Europe, but it's really used a lot in the United States. And the idea here is that by quantifying in some way the health of the insect assemblage and the fish assemblages, they're two separate approaches, we can make inferences about the overall health of the stream or the ecosystem in general in that stream or lake. these are geared towards insects and fishes, but the assumption is that if the insect assemblage is healthy and if the fish assemblage is healthy, then the mussel assemblage ought to be healthy as well. But we don't really know that. And as far as I'm aware, there aren't any methods elsewhere in the world for doing this. There may be some in South America, and if there are, I would like to know about them. But all of these existing methods have problems. One is, as I mentioned, they're specific to a certain region or state or even a specific river or stream, so they can't be applied widely. They often depend on untested assumptions, and I'll talk more about that in a minute. They might require data that people don't typically collect on mussel surveys. For example, the MCAT, it requires estimates of mortality, which can be hard to obtain, and most people don't try to do that in routine mussel surveys. And as I mentioned a minute ago, these insect and fish indices of biotic integrity may not reflect mussel assemblage health. So, it's possible and likely that mussels respond to various environmental factors or human stressors in different ways than insects and fishes, and we'll talk about that a little bit. So to address this lack of objective assessment methods, we developed a thing called the Mussel Assemblage Health Index, MAHI, and these are its benefits. When we developed this, we had these goals in mind. We wanted it to be applicable in streams across eastern North America. So you could compare a stream in Maine or New Hampshire versus a stream in Louisiana, say. We wanted it to be calculated based on commonly collected survey data. And we wanted it to be flexible. We wanted it to be something that you could use with different types of survey data. And we did this because we recognize that everybody, unfortunately, has their own method of monitoring. And these methods, they can be quite different. For example, some people may estimate mussel abundance as density from like quadrats, where they're estimating mussel density per square meter. Other people may estimate mussel abundance based on catch per unit effort, like time searches, number of mussels per hour, or whatever. We also wanted it to be objective to the extent possible and free of these untested assumptions. And so I'll talk about those in just a second. This is available. It was published in June, I think. And here's a citation for it. So here's how we developed MAHI. We came up with a list. Well, I should back up. Things like the indices of biotic integrity and all of these existing mussel indices, they're multimeric indices, which means that they have several different metrics that measure different aspects or characteristics of these assemblages. And the overall score is some combination or average of those metrics. So we came up with a list of 19 candidate metrics that we were going to evaluate. And to come up with this list, we depended a lot on those previous or those existing metrics, like the MCAT. We wanted to really evaluate how those metrics performed. And we came up with a few others of our own, but we can divide these metrics into 5 basic categories. So the first is tolerance and conservation status. So this is a big one where these assumptions, these untested assumptions, come into play. In North America, people have these ideas that some species are tolerant of human stressors or environmental stress, and other species are intolerant or more sensitive. But these categorizations have never really been evaluated objectively, and they can differ. Some people think species A is tolerant and species B is sensitive. Others, if you look at another source, they may think that species A is intolerant. So there's inconsistency. And another thing is that most of the existing probably all of the existing mussel indices in the U.S., they include the number of endangered species in the sample, of species that are listed under the U.S. Endangered Species Act. And the assumption here is kind of like that of tolerance. It's just assumed that these endangered species are more intolerant of basically everything than the non-endangered species. And that assumption has never really been evaluated either. And it doesn't seem like it really holds up very well. So we evaluated these metrics. Similar to this one, there is an idea that certain taxonomic groups or life history groups also respond differently to various types of stress, and that some of them are more tolerant than others, especially like the Anadontinae. People think that species in that tribe tend to be more tolerant of stress, and therefore if you have an assemblage that's dominated by anadontines, it may be in poor health. And same things about host use and life history strategies, etc. And so these are more examples of those untested assumptions. So abundance obviously is something that we want to include in any kind of assessment metric, because the assumption is that more mussels is better. I think that's reasonable. So we looked at density relative to stream size. I'll talk more about that in just a second. And population growth, just whether or not a population is increasing or stable or decreasing. So we looked at several population attributes. As I mentioned, the MCAD developed for the Mississippi River, it looks at mortality. We didn't try to do that because very few mussel surveys have estimates of mortality. So all three of our metrics had to do with recruitment. And this is another thing that you would reasonably want to include in any kind of a metric, because many species in the US are long-lived, and we have many populations that are composed almost exclusively of very old adults, and there's no juveniles, no evidence of recruitment. So this obviously would indicate that the population is unhealthy or declining. And so we looked at recruitment in three different ways, and I'll talk about that in a minute. Another thing that you would reasonably expect would be important in any kind of assessment of the health of an assemblage is richness and diversity. And the assumption, of course, is that higher species richness is indicative of healthier assemblages. And we looked at several different measures of that. We also added a new metric that none of the existing methods included. And this is not just the current species richness, but the proportion of the historical species richness at the site that's still present. So in other words, if we had, if we knew that a site had 10 species historically and there's 2 there now, that proportion would be 0.2. So only 20% of the historical richness is present still. So we developed this list of metrics. Then we compiled survey data from published surveys and from colleagues. We picked 17 streams where people kind of all agreed that these streams were healthy and that the mussel assemblages in the streams were healthy. These are some of the best remaining mussel assemblages in North America. These are in the US and Canada. And then we picked 17 streams where people all agreed that the assemblages are highly degraded or unhealthy. So we just then we calculated all these metrics in each of these assemblages, and we just looked at which ones could discriminate those two groups most effectively. And it turns out that the only ones that discriminated those two groups were these ones that are highlighted. None of these metrics about tolerance or conservation status or taxonomic or life history composition could discriminate these healthy and degraded assemblages. There was a lot of overlap in these metrics. So yeah, these were the metrics that were discriminatory and seemed to be informative. All of the recruitment metrics were informative. We didn't want to have, but they're kind of redundant, and so we just picked the one that was most kind of comprehensive. It's this assemblage recruitment strength. I'll talk about that here in a minute. I'm going to talk about all of these a little bit more to describe what they are, except I won't talk about the proportion of historical richness present. I've already described that, but I'll talk about these other three. So here's the results of how all of these metrics, they discriminated healthy from degraded assemblages very well. There's very little overlap in the values of these metrics. A few of them were, abnormally high or low in some cases, but in general, they did a really good job of discriminating these two kinds of assemblages. And note also that these four metrics, if you include these all as one, they really address four more or less independent aspects of assemblage health. This is the recruitment strength. This is just the abundance. This is population growth, and this is kind of redundant or not quite independent of recruitment, because obviously if a population is growing or increasing, there's going to be more recruitment. But the proportion of historical richness also is basically independent of all these, because you could have a population that has a very low proportion of historical richness present still, but it might be increasing. What species are left might be doing very well. So anyway, so these are the final metrics that we included in MAHI. Again, I just mentioned all these four. So we scaled all these metrics from 0 to 10. 0 means that basically all the mussels are gone. 10 means that everything is great. And the composite score is just the unweighted average of all four of these metrics. So each of these has a score, 0 to 10, and then the composite score also has a score, is on that scale. So just another way of showing you how these composite scores discriminated healthy from degraded streams. Here's the degraded streams. Here's the healthy streams. Here are that composite MAHI score. And you see that only 2 streams overlapped in these 2 categories. We also had a group of about 15 or 20 streams where we and the experts who provided these data to us, they really couldn't make an unequivocal assessment whether it was degraded or healthy, and so we called these undetermined. And as you would expect, those fell kind of intermediate between these 2 groups. And here's just another way of showing this. These shaded boxes are the degraded assemblages. You see that they all have low scores except this one here, and almost all of the healthy streams were higher than the degraded streams. So it seems like that it works pretty well in North America. We also looked at the extent to which Mahi scores reflected expert opinions. So we polled 22 experts and asked them to independently give these streams a score between 0 and 10. And you see that there's a pretty strong positive correlation here between the Mahi scores and the expert scores, and that's good. But you see also that there's a lot of variation here around this line, and that's that subjectivity I mentioned earlier. I mean, everybody has a different point of view and a different way of interpreting what they see. So that's a real benefit of Mahi. It gets rid of that subjectivity. This just shows how widely each expert varied in their scores. Some people, they scored streams consistently high. Other people scored streams consistently low. And again, Mahi, it gets rid of all this subjectivity. And another good thing about Mahi is that we recognize that in some cases, people may not have data to calculate all 4 of these metrics, especially population growth, if you've not been monitoring an assemblage over time. Then you won't have that those historical data that you need to assess whether or not the population is growing or declining. So you can omit one metric and still compute a Mahi score, and it doesn't matter which metric that you omit. They all have kind of a comparable and a relatively minor effect on the overall composite score. So this just shows that by omitting any of these metrics one at a time, you would have an average of about, it would change the score by less than a point. So that's acceptable, we felt like. So I'm going to talk about a few of these, just what they mean, how they're calculated. So the first one is, I said, of course, mussel abundance or density is a fundamental thing that you want to look at. But the problem in North America is that you can't compare big streams versus a small stream because there's a strong correlation in North America of mussel abundance with stream size. We typically find these really dense mussel beds only in large streams where the habitat is real stable, I guess. And you typically find much, much lower abundance in small streams. And that's just a natural thing. It doesn't reflect any kind of human effects. So what we did to account for this was we compiled an independent data set from 51 streams across the eastern part of the US and Canada. And we just calculated the watershed area upstream of the location of the surveys as a surrogate for stream size. And there's a strong positive correlation here. So our metric is just the residual of the expected density based on a stream of that size versus the observed density. So if we have a stream where the natural log of the watershed area is 10, we would expect that the log of mussel density would be about 3.25. If it's above that, it gets a higher score, a positive score. If it's less than that, it gets a negative score. And so that's how we did this. So we also built in a way, I won't go into that, it's described in the paper, but this is all based, of course, on density data from quadrat samples, but you can also use catch per unit effort data if that's all that you have and we have a correction factor there. Well, yeah, but you can only use it if mussel abundance is low, and I won't go into that here, but that's described in the paper. So recruitment, this is kind of complicated. I'm going to sort of gloss over that, but basically we wanted a way to be able to go to look at length data. Most people, when they do a mussel survey, they measure the length of all the individuals. Well, it's not practical to try to age every individual in the field. Sometimes it's inaccurate. and it's time consuming. What you really have to do is to bin section all the shells and age each individual, and obviously that's not feasible. So we wanted to be able to make inferences about recruitment strength just based on the length distributions of those populations. So we used von Bertalanffy growth equations for 92 species in North America. And we found that if you multiply the maximum length that's observed in a population by 0.5, and so half the maximum length, almost all that species, for that value, for almost all species, those individuals are less than 10 years old. So that was our definition of a recruit, was an individual that's less than 10 years old, and that's how we do that. So basically, what it's doing is it's distinguishing these two types of populations. So here on the left is a population where there's a lot of recruitment, and a high percentage of the individuals in the population are less than half of maximum length. And here's the population on the other end of the spectrum. Very few of the individuals, or a low percentage, are less than half of the observed the maximum length. And so this population is going to get a high score for recruitment, and this population is going to get a low score. So we do this individually for each of the species in the data set when the data are, depending on the species, that have an appropriate sample size. But we also wanted to kind of average that over the entire assemblage. And so we also estimate what percentage or what proportion of the species in the assemblage have any kind of evidence of a recruitment, whether or not it's picked up in the survey data. So if the experts considered that 85% of the species in that assemblage are recruiting, we just multiply that by the average of recruitment strength for each species, and that gives us this overall assemblage strength. And we did this because many of the more uncommon species, you just don't have the data to be able to estimate the recruitment strength. So that may not be terribly clear, but I'm trying to just rush through this part of it, but that's described better in the paper also. So population growth is really straightforward. It's just if you've been monitoring a population or an assemblage over time, this is just our metric here is just the slope of the log density. That's all species combined over time. So if you have a positive slope, the assemblage has a higher score on this metric. If you have if there's a negative slope, then there's a lower score to this metric. And you can also use catch per unit effort data here. So now we have these MAHI scores, and we know that they discriminate healthy and degraded assemblages pretty well, but how do they line up with kind of independent measures of habitat quality? Well, in the US, we have some legislation called the Clean Water Act, and a big part of the Clean Water Act is that they monitor water quality and ecosystem health based on the insect and the fish assemblages, and they come up with an assessment of whether that stream is impaired or meeting the standards, which basically means it's healthy. And we see that these MAHI scores, they just span a wide range in all of these categories. And these are sort of intermediate categories, but basically this is an impaired stream and this is a healthy stream as assessed by the Clean Water Act. And we see that in these impaired streams, we have some very, very degraded assemblages, as you'd expect, but we also have a lot of really healthy assemblages. And the same is true on these quote unquote healthy streams. So this type of assessment of habitat quality, it does not inform the health of the mussel assemblage at all. So I talked at the outset about this index of biotic integrity, ways of making inferences about the overall health of the ecosystem in a stream based on the health of the insect and the fish assemblages. And I talked about how the assumption has always been that if the fishes or the insects are healthy, then the mussels are probably healthy. But it turns out that that's not the case at all. There's a weak positive correlation between the health of the fish fauna and the health of the mussel fauna, but you see there's not much explanatory power there at all. There's no correlation at all between the health of the insect fauna and the mussel fauna. So that's important. We have an app that we developed. It's just an Excel spreadsheet that allows you to enter all these data that are needed to calculate each of those metrics. Here are the length data for each species. And it just gives an output here with the score for each of these 4 metrics, and then overall mahi composite score. And it tells you how the stream that you're dealing with here, how it compares with our sort of reference data set of the degraded and healthy streams. And this one is very healthy. It's increasing over time. So then it gives you some details about the recruitment strength and things like that. And there's a link to this in the paper, and I can also, I'll provide a link to this, and we can distribute it to, if you have an email group. So my conclusions on this part of the talk is that mussel declines often, at least in North America and Canada, well, in the United States and Canada, they're often not associated with typical forms of stream impairment. The health of the fish and the insect faunas don't reflect the health of mussel assemblages, not consistently. But the fish and macroinvertebrate insect IBI scores don't always align either. So what that tells us is not surprisingly, the fishes, mussels, and insects, they're all responding differently to different types of stressors. So we're encouraging people in the US to include mahi in addition to fish and insects into bioassessment, because it gives you a more robust and comprehensive picture of the health of the ecosystem in a stream. And we think that mahi can allow an objective assessment of assemblage health over time and over space, different parts of the country. So how could we maybe apply mahi to South America? There are several issues I can imagine, and hopefully we can talk about others that may occur to you all, or maybe ways around these problems. This proportion of historical richness, I can imagine that might be problematic because many areas may not have historical data, so we don't know what species occurred there in the past and may have been lost. In some cases, we can make inferences about that, at least in North America, just based on what we do know in comparable areas. So I don't know about this one, this density relative to stream size. I don't know anything about mussel patterns in South America. I do know that in where I have sampled and worked in Europe, there's really no, well, I've not assessed this, but it seems like there's not a strong positive association between mussel abundance and stream size. And my guess is that will be true in South America also. But if there's not, then that means that you don't have to account for stream size, but you do have to have some sort of a kind of a baseline on which to compare. And so what you could do in a place where there's not a stream size relationship is just compile like a group of reference sites where where you feel like the assemblages are healthy, and that's your baseline. I think that a recruitment strength ought to be applicable, but that will need to be evaluated based on age growth data for species in South America. What the linked frequency distributions typically look like in healthy populations. That's going to depend a lot on lifespan of those populations, and mortality, etc. Population growth, it certainly could be applicable, but you need to have some baseline surveys, some surveys that have happened in the past. But if you have at least 2 points in time, then you can begin to estimate population growth. And the more points in time that you have, the better estimate of this you'll have. So now I'm going to go on to what I mentioned at the outset, the ways of evaluating individual mussel health. These mussels are out there in the streams, they're responding to conditions in the streams. And so ostensibly, we would expect that the responses of these mussels can tell us a lot about the health of the stream. So there's several ways of doing that. In the past, people have tried to look at the physiological health or other measures in wild collected mussels, or they've also caged adult mussels and measured their responses over time. But these two approaches have a couple of disadvantages. One is that in streams that are really highly degraded, there may not be many mussels, and those mussels may have a long and complicated history. They may have been exposed to something bad in the stream that's no longer present, but they still may be showing evidence of that. So we're using an approach where we raise juvenile mussels in a hatchery. We put them out in the stream for about 3 or 4 months, and we look at their responses to the conditions in the stream during that time. This is a great approach because these juveniles are small, they don't take up much space, you generally have a large number of them, and so you can conduct really nicely replicated studies. That's often harder if you're dealing with adult mussels or wild mussels. And importantly, all of these mussels have a common history, so they're comparable to each other. A lot of people use growth as an indicator of health. That's another good thing about juveniles, is that they grow fast, and so you have better power to detect differences in two populations. And some people think that juveniles are more sensitive than adults. We don't really know if that's true always. There are some disadvantages. Of course, you have to have a hatchery facility to raise them. And another big disadvantage of juveniles is because they're immature, they don't reflect any kind of reproductive responses. So if there's a factor in a stream that's negatively affecting reproduction, that won't show up probably in the juveniles. So we put our juveniles in a thing called a silo, which you may have heard about. These have there's a PVC insert that goes inside these things, and you put the juvenile mussels in the insert. And these things have curved sides. Water flows up and over these things, and it pulls water up off the bottom and through. And then, so it brings in food and oxygen and carries away the waste. And so these work really well. They're stable in streams. You generally have to have kind of a hard bottom. You can't put them in mud or pure sand. But we just leave them out for 3 or 4 months. So when I first started doing this, I assumed that we would put these mussels out in some of these really degraded streams and they would just all die. But that doesn't happen. They almost always live, at least for 3 or 4 months. So this is about 23 streams in Kentucky, and about a third of these are these highly degraded streams where almost all the mussels are gone. But they lived in all these streams. Occasionally we have low mortality, but these are just kind of isolated cage effects where a silo gets clogged up or buried. All the streams had at least one silo that had 100% survival. So unfortunately, survival is not informative at all about the health of the stream, at least in the short term. That just tells us, but at least in these streams, there were no acute effects, but there may have been some chronic effects. So growth, on the other hand, is highly variable among streams, almost an order of magnitude or two. So these mussels came out of one of these streams, and these mussels, they came out of one of these streams. They were all exposed for about 3 months. They were all from the same batch of animals raised in the same hatchery. So they're all half siblings at least, but they respond very differently to the conditions in the stream. So that's good. This is something that could be informative. And if you look at the error bars here, these error bars are very narrow, and that's good too, because it means that all of these mussels, and you can see that here, they're all showing more or less a consistent kind of a response to the conditions in those streams. So the most basic thing that people measure in this kind of approach is growth as an indicator of fitness or stress. We can also look at physiological measures or like energy stores. I'm not going to talk about those today, except briefly at the end. Growth is nice because it's easy. It doesn't require any kind of expensive or time-consuming lab procedures. And like I said, it's variable among streams. But the assumption here is that higher growth is always good. And that's what I'm going to really focus on here for the rest of my talk. So, so here are these streams again in Kentucky, lots of variation in growth. So all these streams here have basically lost their mussel fauna almost completely. We calculated these Mahi scores for these streams. And as we expected or assumed, higher growth is associated with streams that have higher Mahi scores or higher mussel assemblage health. So we saw this and we got very excited. We said, okay, maybe growth is a really good indicator of impairment, and maybe what's going on in these streams is that something is interfering with growth, and maybe that's why the mussel assemblages have largely been lost in these streams. However, there's also a stronger correlation between water temperature and growth, and you'd expect this in mussels, of course. And it just so happens by chance that these streams are all cooler. There's also a positive correlation between total organic carbon, if you think about that as a proxy for like total food availability. That's the best that we could do because we still don't really know what mussels eat, and it may differ in different streams. And so we use total organic carbon just as a gross measure of just the productivity or total food availability. Well, also there's a positive correlation here, but it just turns out that these streams, in addition to being cooler, they're less productive. So we could interpret this in at least two ways. One is that this positive association between mussel assemblage health and growth is just an accident. It's a spurious correlation between, because all these streams just happen to be cooler. So it doesn't really reflect assemblage health. It just reflects these kinds of intrinsic relationships between water temperature in foods, and this is meaningless. Or what we could say is that these streams are predisposed to some stressor because they're cooler and less productive. But we don't really know at this point, and that's something we're trying to tease out. But growth is not straightforward to interpret as a measure of health, because there are so many natural factors that influence health, and you have to account for those factors. So the other thing about growth is that you can't just put mussels out in one stream and measure their growth. You have to have some kind of context, as we did here. But the scale is important also. So I showed you in Kentucky, these 23 or so streams, they all have these really interesting kinds of relationships. Well, if you go out in scale, so this is like 90 some, from 50 to 90 some sites across the eastern part of the US. And at this scale, there's no correlation at all between these Mahi scores and growth. There's still a positive correlation between water temperature and growth, and there always will be, but it's much more messy. And there's no correlation between organic carbon and growth at all. So does this mean that that growth is really not an indicator of assemblage health at all, and that food is not important? I don't think so. I think it just means that when you're at such a big scale, there are so many intrinsic differences among these streams, differences in geology, water chemistry, land use, rainfall, all kinds of things, that you have to take all those environmental factors into account before you can begin to make inferences about how anthropogenic factors might be affecting growth, and whether or not growth is an indicator of that. So here's another example of the importance of scale. We're very interested in the US now about the effects, or at least I am, the effects of Asian clams, corbicula, on mussel health. And so we did a study where we put mussels out in about 18, 17 streams for 3 or 4 months, measured their growth and water temperature, and we also estimated corbicula abundance at each of these sites. We also looked at water chemistry, land use, other things. These were the only 2 factors that were strongly correlated with mussel growth. So positive increase of water temperature and a negative association with corbicula. Well, if you put these 2 factors together in a predictive model, it predicts growth almost perfectly. So that was very interesting to us, and we thought, well, maybe we're onto something here about growth and corbicula. But now this is another example of scale. If you go out to the eastern part of the US, as a whole, from Minnesota to Alabama, there's no correlation at all between corbicula and growth. So again, does this mean that there's no effect of corbicula? Well, I don't think so. It just means that there's too many, there's a lot of environmental factors that differ among all these streams, and you have to take all those things into account before you can really look at any kind of the potential relationship between corbicula and growth. So another way that we can look at growth is by looking at the growth history in the shells. We did that for 2 species in the Illinois River in the US over like the last 1,000 years. These are mussels, or these were shells in an archaeological deposit, and these were historical shells out of a museum collections, and these are contemporary mussels. We found that the growth rate is increasing gradually over time, and they get bigger. in both species. So all this is interesting, but what does it mean for fitness? There's definitely a response of the environment, or maybe of corbicula, but so what is it? What does it mean for fitness? Does it necessarily mean that these animals are less healthy than these animals here? Let's talk about this. So let me go back for a minute. So here, remember, our assumption always was, that these animals are healthier, and pretty much everyone else who does these kinds of studies, they go into it with that assumption that higher growth is better. Well, in this study, we kind of interpreted this increase in growth as a bad thing. We interpreted it as a reflection of nutrient loading in the stream, or eutrophication. We said it's a bad thing. We said that we'd like to see growth more like this. So let's talk about the validity of this assumption that higher growth is always good. Well, one problem with it is that we know that there's a trade-off between growth and lifespan. These are 90-some species and populations in North America, and we see that there's a strong exponential decrease in growth rate with lifespan. So if the population is growing faster, it's not going to live as long. Another problem is that this assumption that higher growth is always good is at odds with other predictions that we have. So thinking about climate change, everyone thinks that climate change is going to negatively affect mussels. And they may be right, but if our assumption is that higher growth is always good, well, if the temperature increases over time, then mussels are probably going to be growing faster. So does that mean that we should be pumping out as many fossil fuel waste into the environment as we can in order to try to raise the temperature? Probably not. People are also concerned about excessive nutrients and eutrophication in streams. If more food or more production means higher growth, then we shouldn't be concerned about this. It means that nutrient loading is not bad. So obviously that's not true. So these are two examples of how this assumption that higher growth is always good is sort of unreasonable. The other thing is that we interpret growth, as I've alluded to, based on whatever preconceptions that we have going into a study. So let me see what's, hold on a second. So in those silo studies I described, our preconception was that higher growth is good and low growth is an indicator of compromised health. Well, in this study, we made just the exact opposite interpretation. We interpreted this as that this higher growth is bad, and that this is more desirable because it reflects a less nutrient-enriched habitat. So there's a lot of things that we have to think about, a lot of factors to account for, and we have to be kind of clear and objective about our assumptions about whether growth is good or bad. So just some conclusions. These in situ exposures of juveniles and probably adults, they're uninformative in the short term for survival. And growth, I think, has the potential to be informative, and I think that it is, but it's really difficult to interpret because you've got to have context, you got to think about the scale, and you have to take into account these intrinsic factors that are always going to influence growth, like temperature and food, etc. So I do think that in situ exposures are going to be effective. We're looking at physiological measures or pathogen burdens, etc. We're just now beginning to to evaluate these kinds of approaches, but I think these are often free of a lot of the problems that growth might have. And just to reiterate again, if you're doing any kind of measurement of growth, we need to evaluate this assumption that higher growth is always good. And that's all I have. Here's again a citation to that paper about mahi. And at that point, I guess I can take some questions or comments.